This
Zhou from
conference will
NIH
now
and
be
HLBI,
recorded.
and I will be your moderator today. Dr. Okawa is the professor of medicine at Harvard Medical School. She is the director of Vascular Biology Program Center for Interdisciplinary Sciences. Dr. Okawa is also the founding director of the Heart Valve Translational Research Program, as well as the associate head of section of CV Life Sciences at Brigham and Women's Hospital. Dr. Okawa's research interest focuses on the development of new therapies to prevent, treat, and cure calcific aortic valve stenosis, most recently using multi-omics and network medicine approaches. Her work on inflammation-dependent mechanisms of CV calcification has led to the discovery of exosomes as the precursor of microcalcification. Dr. Aikawa is the recipient of numerous accolades, including more than 200 publications, as well as multiple editorial positions. She is also the vice chair, has been the vice chair for the Women's Leadership Committee of the AHA Council on ATBB, as well as a past president of the International Society for Applied Cardiovascular Biology. Dr. Piotr Slomka is the Director of Innovation in Imaging, Artificial Intelligence and Medicine Program and Professor of Medicine at Sinai Medical Center in Los Angeles. He's also the Professor of Medicine at the David Giffen School of Medicine at UCLA. Dr. Slomka is the Principal Investigator of two NIH-NHLBI R01 grants and is the author of more than 320 publications, including 17 book chapters. Dr. Slomka's research interests include improving the diagnosis and risk assessment of heart disease by developing novel software methods and image processing algorithms. Dr. Slomka is the recipient of numerous awards, including the Herman Blumgart Award from the SNMMI, as well as the Zira Beller Award for Best Reviewer for the Journal of Nuclear Cardiology. He's also the recipient of the JNC Best Paper winner in the Basic Technical category. A little bit more about Dr. Slomka's research areas. His research focuses part one on high performance automated system for analysis of fast cardiac SPECT. He is focused on using methods to innovative methods for automated analysis of nuclear cardiology data using novel algorithms and machine learning techniques. Dr. Slomka is also investigating using integrated analysis of coronary anatomy and biology using 18F fluoride PET and CTA. He's also using this to optimize the measurement of disease activity of coronary atherosclerosis using 18F fluoride PET CTA imaging. Dr. Martin Lingby Lassen is a postdoctoral fellow in Dr. Slomka's lab at Cedar Sinai. He received his PhD in medical physics from the Medical University of Vienna. His research focus is on improving quantitative PET imaging through motion correction, tracer kinetics, and improved attenuation correction. Dr. Lassen is the recipient of the Young Investigator Award presenter at SMMMI in both 2019 as well as 2020. Dr. Lassen is also the recipient of the Olavi Mendel Award in 2020. Just a public service announcement, Dr. Slomka actually has several job openings in his lab at Cedar Sinai, including positions for postdoctoral scientists, project scientists, as well as research software engineer. If anyone is interested in these positions, please don't hesitate to send a CV to Dr. Slomka at the email address listed here. Dr. Zahi Fayad is the Lucy M. G. Moses Professor of Medical Imaging and Bioengineering at the Icahn School of Medicine at Mount Sinai. He is the Professor of Radiology, the Vice Chair for Research, and Professor of Medicine and Cardiology. Dr. Fayad is also the founding director of Biomedical Engineering and the Imaging Institute. Dr. Fayad's research interests include detecting and preventing cardiovascular disease with many seminal contributions in multimodality biomedical engineering, including MR, CT, PET, and PET-MR, and also nanomedicine. Dr. Fayad specifically is interested in looking at the effect of stress on the immune system and cardiovascular disease. Dr. Fayad has more than 300 peer-reviewed articles, as well as serving as on multiple editorial boards. Dr. Fayad is the recipient of multiple awards, including the Outstanding Teacher Award from the International Society of MR in Medicine, as well as the Distinguished Investigator Award from the Academy of Radiology Research. Just as a reminder, next week's session will take place Wednesday, June 3rd from 11 a.m. to 12 p.m. And the topic of that session will be transthyretin amyloidosis, featuring a didactic lecture by Dr. Jeffrey Kelly from the Scripps Research Institute, followed by a research presentation from the laboratory of Dr. Prem Soman from the University of Pittsburgh. The discussant for that session will be Dr. Edward Miller from Yale University School of Medicine, and the moderator for that session will be Dr. Sarah Cuddy from Harvard Medical School. Just as a reminder, the deadlines for research awards and grants for ASNC is upcoming in June, and also any information can be found through email on the email below. So without further ado, I'm going to turn the mic over to Dr. Okawa, and she can start our session. Zhou, would you have Dr. Okawa as the presenter?
All right. Good morning, everybody. First of all, I would like to thank the organizers of this meeting for inviting me for this exciting seminar series. I think it's excellent idea to link basic science with clinical research. Today, I'm going to talk about calcification, basic biology for imagers. I have no disclosures. So first of all, we need to understand why calcification matters. And calcification matters because arterial microcalcification may cause plaque rupture at interface areas between high and low density tissue, which can cause heart attacks. And aortic valve macrocalcification, on the other hand, impair the movement of the valve leaflets, resulting in valve stenosis and heart failure. However, there are no therapeutic options available to prevent or treat calcification. And also, there is a lack of clinical imaging modalities to detect calcification early. So today, I'm going to focus on arterial microcalcification. Let me find the laser pointer. Yeah, this one. All right. And just to be with you on the same page, I would like to remind you that there are two types of calcification in atherosclerotic lesions. The microcalcifications, when they are located in thin atherosclerotic plaque, can cause plaque instability and potentially rupture. And macrocalcification, which are usually embedded within the body of atherosclerotic lesions, can potentially stabilize the plaque. So this is a nice schematic by Linda Dimmer, which demonstrates relationship of calcification and surface area, which can explain the biphasic clinical risk associated with coronary calcification. As you can see here, the larger calcification area, the higher plaque rupture risk. And in addition to surface area, location, size, and shape of microcalcification can contribute to plaque instability. More than a decade ago, we published a series of these papers where we demonstrated that molecular imaging can detect early calcification. We showed that near-infrared molecular imaging detects early stages of calcification, which are undetectable by routine histology, such as Van Kassa, and routine imaging modalities, such as CT scan. You can see much more calcified cells detected by near-infrared as compared to just 2 cells detected by Van Kassa. Same is here. And we also, for the first time, were able to demonstrate presence of microcalcifications in atherosclerotic lesions of mouse model of atherosclerosis. In addition to identifying microcalcifications, using infrared imaging, we also co-localized inflammation with early calcification, as shown here, and this is a very nice correlation of signal from macrophages and osteogenic activity. All these imaging studies allowed us to propose 3 stages of inflammation-detected calcification, which we identified by in vivo molecular imaging. We now propose that calcification starts with the stage of inflammation, it was shown here as a green fluorescence, when inflammation precedes calcification. In the stage of propagation, both processes develop within close proximity simultaneously. You can see correlation of inflammation and calcification in atherosclerotic lesion. This stage also characterizes by abundant release of calcifying extracellular vesicles from macrophages and smooth muscle cells. And this is a topic of my research, which I will explain later. Potentially, this stage is reversible. However, advanced calcification, which shown here as red fluorescence, characterizes by prominent mineralization and reduction of inflammation. Potentially, this stage is irreversible. But we always, so we were very excited that after 5 years, when we published our studies in mouse model of atherosclerosis by molecular imaging and detection of calcification and co-localization of calcification and inflammation, our colleagues from MGH reported first-in-man evidence that local arterial inflammation precedes subsequent calcification in the same location using longitudinal FDG PET-CT studies. So it was very exciting. But we always need to remember that only 10% of calcification is developed through osteogenic activity, and the other 90% of cases happen by unknown mechanisms. Therefore, my group of these talented investigators, talented postdocs, started to look into alternative mechanisms of calcification via the release of calcifying extracellular vesicles. So our current hypothesis is that extracellular vesicles generate microcalcifications, as shown here, and this then microcalcification generate high stress concentrations around the poles. And if this microcalcification located in a thin fibrous cap, that can cause plaque rupture. But what are extracellular vesicles? In our case, extracellular vesicles are approximately 100 to 300 nanometers. They are perfect communicators between cell and extracellular matrix, and generally they keep homeostasis of the cells. However, in pathological conditions such as atherosclerosis or chronic kidney disease or diabetes, vesicles also gain pathological cargoes, such as annexins or high levels of calcium phosphate or alkaline phosphatase, and I will touch base on this a little bit later. And these pathological vesicles can contribute to osteogenic differentiation of smooth muscle cells to osteogenic cells or contribute to calcification of extracellular matrix. As pathologist by training, I often look at the electron microscopy images, and I could see many different types of extracellular vesicles within the tissue and within the cells, which grow in osteogenic media. So you can see multivesicular bodies in atherosclerotic lesions. You can also sometimes see this complex of vesicles released outside the cell, and they're covered by the same basic membrane. Eventually, this membrane will be resolved and vesicles will be released in extracellular matrix. This is a macrophage stained with CD68 immunogold antibody, shown here this, as a black dots. And you can see this macrophage releases extracellular vesicles. And these vesicles have double membrane, suggesting that they were just released from the cell. From parental cell, and they can nucleate hydroxyapatite crystals. And here we can see that vesicles can contribute to calcification of collagen fibers. But as technology progresses, we see more and more details in atherosclerotic lesions. So this is a density-dependent scanning electron microscopy. Here is advanced calcification, or we call it macrocalcification, shown here by orange. And we notice that these macrocalcifications in atherosclerotic lesions always surrounded by microcalcification and calcifying extracellular vesicles. Every particle which is larger than 1 micron in size, we call microcalcification, and below this size, calcifying extracellular vesicles. And we know that these particles are calcifying particles because elemental analysis showed high levels of calcium and phosphate. So in 2013, we published this small review article where, for the first time, we proposed that both macrophages and smooth muscle cells in the fibrous cap of atherosclerotic lesions can release calcifying extracellular vesicles. Some of those vesicles are very small, even below 100 nanometers, so they can go easily through endothelial barrier and join the pool of circulating microparticles. But some of those vesicles are larger in size, can be trapped between collagen fibers and become microcalcification. So my early imaging studies, when I was trained in Ralph Weiss-Leder's group in 2005, 2010, 2009, something like that. We looked at the calcification of atherosclerotic plaques. So this is an in vivo imaging of carotid artery of APO in knockout mouse. And again, you can see strong localization of inflammation, which is green, with calcification, which is red here, and the colocalized signal shown here as yellow. So this suggested to us the hypothesis that macrophage may contribute directly to microcalcification formation. And we published this report in 2013 where we showed that macrophage derived extracellular vesicles contribute to calcification through the formation of S100A9 annexin 5 and phosphatidylserine complex with hydroxyapatite. So we now extended the studies to in vivo research, and this is an image of atherosclerotic plaques in diabetic mice. We just published this paper. Actually, it was just accepted to ATVB after very extensive reviews. And you can see very clearly that when we injected our mice, diabetic mice, with macrophage-targeted SIS-100 A9 lipid nanoparticles, they could dramatically inhibit calcification in atherosclerotic lesions as compared to diabetic mice calcification shown here as red. But today I'm going to focus on calcification of smooth muscle cells. If you have smooth muscle cells which grow in osteogenic environment, they start to produce these calcifying particles, which we can detect by near-infrared calcium tracer, so-called OsteoSense, the same one we're using in our in vivo studies. In addition to that, cells also produce large amount of collagen. So-called fibrocalcific response of smooth muscle cells to osteogenic environment. Very important observation. However, more important observation of this paper was how macro and micro calcifications form. What we noticed that if our particles aggregate below dense collagen fibers, they form macrocalcifications. But if your collagen fibers are not so dense, vesicles could go in between, or particles can go in between those collagen fibers and remain microcalcification. So this is evidence of the same thing, but now in human plaque and mouse atherosclerotic lesion. You can see collagen fibers here are green, vesicles are orange, and they stuck between collagen fibers and forced to aggregate. Same is in mouse plaque. So we were wondering why our vesicles aggregate with each other. What makes them aggregate? Because we think that this process of aggregation of the vesicles are very important for formation of microcalcification. So what we did, we isolated vesicles from calcifying smooth muscle cells and performed proteomics analysis of those vesicles. And we found, as shown here, this volcano plot, that annexin 1, which is calcium binding protein, is one of the most abundant proteins expressed on this or present in those vesicles. We then did quadruple staining of annexin 1, is shown here as red. Calcification, you see those vesicles are in white, which you can see stuck between collagen fibers, which are green here. So you could see strong colocalization of annexin 1 with calcification. Electron microscopy also demonstrated that annexin 1 particles are sitting on the surface of calcifying extracellular vesicles. So here is single cell extracellular vesicle analysis, which captures annexin 1 positive extracellular vesicles, as they're shown here as red. You see the predominant number of the vesicles isolated from calcifying cells are annexin 1 positive, as shown here by quantification. So now the question was whether annexin 1 is indeed directly contributes to aggregation of the vesicles. So Max Rogers, who was first author of this paper, created artificial vesicles from a phosphatidylserine membrane, phosphatidylserine lipids, and he added in that culture of the vesicles annexin 1 GFP labeled, annexin 1 labeled with GFP. And he noticed that when annexin 1 is located to the surface of the vesicles tend to aggregate with each other. Interestingly, when he added annexin 1 neutralized antibody, they inhibited vesicle aggregation. Two cultures of two assays for calcification. One is smooth muscle cells grown in osteogenic environment, and another one is smooth muscle cell derived extracellular vesicles grown on collagen fibers, showed dramatic reduction of calcification after annexin 1 silencing. So those studies suggest that membrane of extracellular vesicles contain tethering proteins that induce vesicle aggregation and promote calcification. However, these processes of vesicle aggregation is impossible to visualize in vivo in human or mice, even in mice, because all these processes lay below the resolution of current imaging modalities. So we needed to come up with some idea to have a platform which could allow us to look at the kinetic information of microcalcification. And Josh Hutchison, who was first author of this paper, proposed the idea to have collagen fibers, which he seeded with calcifying extracellular vesicles, which were isolated from calcifying smooth muscle cells. And we were able to recapitulate perfectly early and advanced stages of calcification in atherosclerotic plaque. And of course, this process of advanced calcification happened to human maybe within decades or at least many years, but we could see this similar aggregation of the vesicles and formation of advanced calcification within a few days. This platform also allowed us to visualize formation of the microcalcification on the level of the one single vesicles, as shown here. This vesicle is approximately 200-300 nanometers, and you can see that vesicles start to aggregate with each other when they're still not calcified. And eventually, calcification ring would form, and microcalcification would form. And we were able to detect it with high-resolution structure elimination microscopy. So in this paper, which we published in Nature Materials several years ago, we demonstrated that, or we proposed rather, that cells release calcifying extracellular vesicles. They aggregate with each other, merge, nucleate hydroxyapatite, and become microcalcifications. So these studies also allowed us to propose unifying hypotheses. The first hypothesis was proposed by Peter Libby and his colleagues more than 2 decades ago, which suggested that collagen degradation can contribute to plaque destabilization. But we now propose that in addition to collagen degradation, you have to have these impurities in form of microcalcification within collagen fibers, and if they have high stress concentrations, they can contribute to plaque destabilization. On the other hand, if you have intact collagen fibers, vesicles will keep aggregating below these collagen fibers and form macrocalcification, which could contribute to plaque stabilization. So, well, now we know what happened to the vesicles when they released outside the cells. They aggregate, merge, nucleate hydroxyapatite, become microcalcifications. But what make those vesicles to be calcification prone? It was still unknown. But in the same year, we published a paper in 2016 in GCI, where we showed what happened within the cells and how these calcification prone vesicles form. Apparently, it happened that in trans-Golgi network, serine kinases, phosphorylate certillin. Certillin is a transporting protein. It's been known for many years to transport cholesterol within the cells. But now we found new biology of certillin and found that it also can bind to active alkaline phosphatase within the Golgi and take this alkaline phosphatase into recycling endosome via RAB11 pathway. And then this complex merged with plasma membrane rich in caveolin-1 and vesicles which contains high levels of alkaline phosphatase released outside the cells. Alkaline phosphatase, or TNAP, is very important key protein for calcification. So we were excited. We identified that phosphorylation contributes to TNAP trafficking into the extracellular vesicles, but we also realized that dimerization is also very important for certillin itself to travel into the vesicles. And this paper was published 2 years ago in JBC. So we then validated our in vivo studies in in vitro studies in 2 in vivo models. One is double knockout certillin deficient mice on LDL receptor background, and another is certillin deficient mice injected with PCSK9. And using molecular imaging, which we validated or confirmed by histology, we demonstrated reduction of osteosense signal, reduction of calcification, and alkaline phosphatase activity in both models. However, probably the more exciting and more important observation was that we found that serum certillin associates with aortic calcification and cardiovascular risk in humans, independently from C-reactive protein and LDL cholesterol. So all the studies in combination allowed us propose our own model of cell-derived extracellular vesicle microcalcification formation. We propose that osteogenic condition in mitochondria activate alkaline phosphatase. Then alkaline phosphatase is loaded to extracellular vesicles via certillin to make them calcification prone. Then annexin 1 promotes aggregation of the vesicles. Annexin 1 shown here is green. And then vesicles are trapped within collagen fibers. And finally, aggregated vesicles nucleate hydroxyapatite and become microcalcifications. So in conclusion, we showed that near-infrared molecular imaging identified for the first time microcalcifications in atherosclerotic lesions of mice, that molecular imaging showed in vivo that inflammation precedes calcification. It was very important observation for the inflammation-dependent mechanisms of calcification. We found that both macrophages and smooth muscle cells release extracellular vesicles, and these extracellular vesicles are precursors or building blocks of microcalcifications. We also demonstrated that phosphorylated sortilin is transport of alkaline phosphatase into extracellular vesicles, which is very important to make them calcification prone. And we think that sortilin is a potential biomarker of arterial calcification, as we showed in human studies. So at the end, I would like to thank my wonderful postdocs who contributed enormously in these studies, and also my funding sources from NIH, Harvard Catalyst, and Cola Company. Thank you very much for your attention, and I'm very happy to answer questions. Thank you.
Okay, great. So this is Zahi Fayad. I will be taking the questions, reading them from the chat. If you have any questions, please send them over in the chat boxes. Wonderful work and presentation, really summarizing this whole idea of microcalcification and really mechanistically what is this all about. Elena, let me start with a question before I take some from the audience here. I was intrigued and pleased to see your approach of trying to therapeutically target the microcalcification with the nanoparticle probe that you tested in diabetic mice. So I have two questions. Number one, what is the frequency of microcalcification in this animal model? And maybe you can talk a little bit about that. And then the second question, can you give a little bit of a detail on the platform that you use and the biological target that you went after?
Yeah, well, thank you very much, Zahi, for your questions, very important questions. So first of all, question about diabetic model and how often we see calcification in this model. So diabetic models are characterized by accelerated calcification. And at the same time, in addition to calcification or microcalcification formation in diabetic model, you can see also abundant inflammation. So these two processes co-localized and coexist in the same atherosclerotic lesion. So you could see abundant macrophages mixed up with microcalcifications. So that suggests that probably a calcification in these mice released not by smooth muscle cells, which we often see in atherosclerotic in culture conditions, but by macrophages. And that was our hypothesis in that study. Therefore, we decided to target macrophages in this model by introducing the lipid nanoparticles, which contained a macrophage, specifically macrophage targeted S100A9 siRNA. And it was actually amazing because it seems that when we kill all these macrophages with S100A9 biosilins and S100A9, we didn't see microcalcification formation anymore. And yeah, so that was our strategy.
Okay, great. So let me take questions posed by the audience. First question is about the study associating serum sortilin with plaque, sodium fluoride, or FDG PET uptake. Do you have any idea if there's any human data looking at this marker, I guess serum marker versus an imaging marker?
No, I don't think any study like this has been done yet, and I think it's a very good idea to combine both. I think the imaging biomarker, right, would be perfect to combine with real biomarker like in the blood, and look at the both or predict rather calcification formation or early calcification in our patients. Because we really believe that when you have advanced calcification may be not so harmful to patient, but this very small microcalcification, when they're located in the thin fibrous cap, they can cause plaque instability.
So how easily is it to measure serum sortilin in human? And how validated is that marker? Any lab can do this?
I think so. I think we've done that. So I think any lab can do it as well. So you can use ELISA, and you can measure sortilin in the blood. We had, I don't remember how many, probably 800 samples or something like that. And we measured one or two person, I think Claudia Ghosh, who was running this study, and technician was measuring this blood together. Okay. So I think it's feasible. It's very feasible.
Another question is about the collagen synthesis in the ER. What role does it play for calcification?
Collagen synthesis in calcification, very good question. You notice something very important. So interestingly, calcification, microcalcification need to aggregate, and I think they need to have a space for or niche for aggregation. And if they find this kind of niche within broken collagen fibers, they will keep accumulating within that niche, because otherwise they will be moving in interstitium of atherosclerotic plaque. You can actually, because plaque is very viscous, right? And vesicles are easily can move within like lipid particles and between macrophages, between cells, until they stuck, until they find something which prevent their movement anymore. And then if they stuck within this kind of like voids of which calcification, broken fibers, broken fibers of collagen form, they will form this microcalcification by aggregation there.
Okay. Do we have time for more questions? I'm asking the moderator because I didn't keep time on my watch here.
We actually have to move on to the next
we have to move on. Okay.
Yes.
All right. Elena, thank you so much. This was really wonderful and extremely well received from the messages I also got on the chat. Thank you again.
Thank you. Bye.
I guess we can move on to the next lecture. I'm not the moderator, so I'm not sure who's introducing this speaker.
Yeah. Dr. Lesson, please start when you're ready.
Okay, thanks a lot. Thanks a lot for giving me the opportunity to present our work on the technical advancements of sodium fluoride PET imaging, which I have also expanded to also include rubidium imaging, where we face some of the same challenges as we do for sodium fluoride PET imaging. During today's talk, I would cover several different topics, but I will first start with sodium fluoride imaging and then afterwards transition into the rubidium scans. So, as already been explained in the very nice talk just before, and also in the talk last week and last Thursday, we know that cardiovascular diseases is the leading cause of death worldwide, and currently in the nuclear medicine and also in molecular imaging societies, we're trying to identify different tracers that we can use to identify which lesions that might turn culprit. Here, sodium fluoride is one of those tracers that have shown tremendous potential to identify the culprit lesion. This was first shown in the Zhou paper published in the Lancet in 2014. So in very brief, as already just discussed, sodium fluoride is attaching to the microcalcification that is occurring on the plaques in the cardiovascular tree. And this uptake itself helps us to differentiate between lesions that are turning into microcalcification and the ones that can rupture and undergo thrombosis. So using the sodium fluoride PET imaging has several disadvantages. First of all, we speak of moderate target to background ratios, as shown here, and was also shown in the paper by Dr. Zahi from the Edinburgh group, where we can see that targeted background ratios are roughly around 1.2, 1.5 for the culprit lesions, whereas non-culprit lesions are slightly reduced. Nevertheless, nevertheless, we can differentiate between the two, both the culprit and non-culprit lesions. In addition, we have significant blurring due to motion during the scans. That can be either cardiac motion, respiratory motion, or patient repositioning events. And finally, using the conventional analysis, we have low, high noise in images due to low count statistics, as shown here, where we can see that the lesion is standing out from the background activity, but it's difficult to differentiate between lesion uptake and background activity. So here in our center at Cedar, we have tried to evaluate different techniques to identify which lesions that are culprit and how to improve the quantification. In the first paper I'm going to present, we evaluated a 3-hour delayed imaging protocol in comparison to a standard 1-hour imaging protocol. In the studies that is acquired during the Diamond and Prefer studies, we rely on injection to scan delay protocols of 60 minutes, where the patients are injected with roughly 250 megabecquerel sodium fluoride, and followed by rest in calm conditions for 60 minutes. In our paper published in JNM in 2019 by Dr. Krasinski, we evaluated what impact it does have to extend the injection to scan delay to 3 hours. We observed there was several different improvements. First of all, and the most important was that we observed that the blood pull activity as shown here using 1 hour injection scan delay diminished when we extended the injection scan delay. More importantly, we also saw that we could identify that the lesions is easier to identify in the 3 hour using the 3 hour injection to scan delay protocol rather than 1 hour injection to scan delay protocol. This diminishing of the background activity led to significant improvements in cyclic background ratios, and here it's of importance to notice that even though we have increased tight background ratios, we observe that the HRV or the uptake in the lesion itself is quite homogeneous throughout the 1 hour and 3 hour imaging protocols. Using this information, we try to identify in a paper published in EGNM last year what impact it does to have to improve by introducing a blood pool clearance correction. So using the same data as presented by Dr. Krasinski and colleagues, we were able to show that that background blood pool activity deteriorates with somewhat an exponential function, which can be shown here. This becomes extremely important when we start speaking of using 1 hour injection to scan delays, where we can see that there's some variation, even though we should use a normalized protocol. This becomes even more evident when we started analyzing 20 patients who underwent test-retest scan as a part of the Diamond study, where we observed that injection to scan delays could vary from anywhere between 59 minutes and all the way up to 101 minutes. More importantly, we also saw that there was significant changes in the test-retest protocol, which might affect the quantification. So we try to evaluate the impact of doing this simple background blood flow correction technique on endocytic images, where we were able to show that by introducing this simple correction, we were able to improve the retest variability, such that we improved the retest variability by more than 15%, thus suggesting that these injection scan delays have significant impact on quantification. Unfortunately, we have several other problems, as already discussed. We also have problems with motion during the scan, which I will cover in the next paper, which was published in JNM in 2019. Again, coming back to the Diamond and Prefer imaging protocols, we can see that we rely on 30 minutes acquisition protocols, which have some disadvantages. First of all, during the very long acquisitions, we have problems with both coronary contraction or cardiac contraction, which translate the coronary arteries of up to 26 millimeters, as was shown in the paper by Dr. Robreau from our team in the JNM paper of 2016. In our JNC paper from 2019, we were able to show that respiratory motion might translate the heart as much as up to 31 millimeters. And finally, in the current paper I'm presenting right now, we were able to show that patient motion could translate the heart of up to 60 millimeters. Unfortunately, there's no easy way to detect when patient reposition during the scans. So in the current study, we evaluated we extracted the information of patient repositioning events using an this node-based approach only, meaning we only use the PET raw data. By sampling the PET raw data in very short time points of 200 milliseconds, as shown here, we were able to identify when the patient repositioned by using a center of mass approach or analysis of these 3D sinograms. In doing so, we were able to obtain a patient reposition curve, as shown here, where we were able to identify when the patient repositioned. In the current study, we evaluated how much impact the patient motion has on the data by using endoscopic images, as shown here. Here is shown one case example where you can see how the patient motion during the scans affect both the contrast in the images, but also the target background ratios and other quantitative measures. We can see that following the motion compensation, that the lesion is quite evident in the lesion when compared to before. In the given case, the patient had two repositioning events during the scan, one where the patient was lowering the arms after 3 minutes and then elevating the arms again, thus suggesting that motion can occur at any time during the scans. In general, when speaking of these motion patterns, and when corrected for them, we were able to identify that in lesions moved by more than 10 millimeters as a function of the repositioning events, we had improvements in target to background ratios of roughly 15% and significantly improved contrast in the images. Unfortunately, patient motion is not the only problem we face. We also have problems with both cardiac and respiratory motion, as already mentioned. And in our EGNM paper from 2019, we evaluated how to expand the patient motion detection and correction to also include corrections for cardiac and respiratory motion. So the data was acquired using a standard ECG gating approach, using a 3-lead electrocardiogram as shown here, where we used 4 ECG gates. We also expanded the data-driven patient detection, motion detection approach to also include detection of respiratory events by only evaluating data obtained from the diaphragm region. By doing this, we obtained a complex series of motion patterns consisting of both external markers and data-driven markers, as shown here, which were consistent throughout the scans. To compensate for this motion, in this study, we evaluated the feasibility of using a triple-gated reconstruction, where we performed a dual-gated reconstruction per patient event, meaning that we used 4 ECG gates and 4 respiratory phases per patient repositioning event. Introducing corrections for these, using this triple motion compensation technique, we were able to see that we could improve the targeted background ratios and contrast in the images using a single scan as shown here. More importantly, we were also able to show that the variability between 2 scans as shown here in terms of contrast was significantly improved using the triple motion compensation technique in comparison to the standard techniques. Also of high importance, we were able to show that these motion compensation techniques also improved the target background assessments in comparison to ECG motion compensated using ECG motion compensated data or respiratory motion compensated data. Of extreme importance in context of translating sodium fluoride into being a clinical marker, we also need to be able to have high reproducibility. In the same study, we were also able to show that test-retest reproducibility using a standard endoscopic assessment led to relatively poor coefficient of reproducibility. However, by introducing this triple motion compensation plus the blood flow correction, as already discussed, we were able to more than half the test-retest variability, and thus suggesting that motion compensation and blood flow correction really improves the quantitative assessment of sodium fluoride PET data. These corrections become even more important when you start evaluating how to assess and quantify the lesion uptake. Until now, we have been basing all our assessments on targeted background ratios in a single lesion assessment as shown here, where we normalize the maximum HRV in the lesion to the background activity obtained in the right atrium. However, in our 2020 EGNM paper, we evaluated the possibility of expanding this to evaluate the uptake in the entire coronary tree, either per vessel or in the entire coronary tree, where motion compensation might be even more important. Finally, now I would like to go into verbatim imaging that is not really related to the sodium fluoride imaging, but still we have some of the same problems when discussing myocardial blood flow assessments using verbatim data. So the first paper I'm going to present was published in JNC in 2019 by Dr. Zahi and myself, where we evaluated the short-term test-retest reproducibility of myocardial blood flow using verbatim and how the motion compensation and the use of two different input functions affected the myocardial blood flow assessments. In this study, we included a total of 21 patients in a test-retest protocol, where the patients were injected with roughly 1 gigabecquerel of verbatim. Both scans were performed at rest and only a few minutes apart, as shown here. In the study, we performed 3 different subgroup assessments, one using the standard assessments as implemented currently in PurePad and also in all other clinical softwares, where the myocardial blood flow is evaluated using the left ventricular input function without any motion compensation of the image during the scan. We expanded this protocol to also include motion compensation, as shown here. And finally, we compared the motion compensated images obtained using the left ventricular input function and the left atrial input function. From this study, we were able to conclude 3 important things. First of all, we were able to conclude that test-retest assessment using in a rest scan assessment had relatively acceptable test-retest variability, as shown here. However, introducing motion compensation when deemed necessary reduced the variability between the scans. Importantly, in this context was also that this motion compensation was performed by 2 independent observers, and we were able to show that the inter-observer variability was much less than the in-between variability. The third and important thing we were able to conclude was that there's a bias when obtaining myocardial blood flow using left ventricular input function and left atrial input function. Unfortunately, in the given study, we were not able to conclude which one is most accurate, as we didn't have any outcome data for these patients. Also important in context of the myocardial blood flow assessment and the motion compensation is how many frames we need to perform the motion compensation as this is a tedious and time-consuming process, especially when we speak of multiple myocardial blood flow assessments per day. So in the same study, we evaluated the feasibility of using data from only the first 2 minutes comparing to perform motion compensation of the full data set. We were able to show that by performing motion compensation for the first 2 minutes, we were able to obtain myocardial blood flow assessments similar to the ones obtained when we perform motion compensation on the full data set, thus suggesting that we can rely on the myocardial blood flow assessment even when only performing motion compensation in the initial phase of the scans. In the last presentation or paper I'm going to present today, I would like to discuss what another problem that is also affecting myocardial blood flow, and namely the injection protocols that are currently suggested. Currently, SNM and ASNC recommended guidelines suggest injections of 30 to 40 millicurie or approximately 1.5 gigabecquerel of sodium fluoride of iridium, which might affect the quantification of the myocardial blood flow. In this study, we included 20 patients, again scanned in a test-retest protocol performed in rest setting only, where the patients were injected with, in the first scan, 1250 megabecquerel as the first scan, and then in the second scan, the patient had an injection of 660 megabecquerel of sodium fluoride. The data were evaluated on 3 different parameters to assess whether PET system saturation was present or not. The first and most important was in assessment of the input functions as shown here in the first column. Evaluating the data, we can see that we have 2 different features. Either we have a sharp peak or we have a blunt peak as shown in A through C. Once we have a sharp peak, we don't expect any saturation of the PET system, whereas the blunt peak might suggest PET system saturation. By evaluating the first pass bolus in the left ventricle, as shown here, we can differentiate again between saturated data set and non-saturated data set. When homogeneous uptake patterns are shown here, is present, we don't suspect any PET system saturation, whereas speckle noise, as shown here in A and B, might indicate PET system saturation. Finally, we can conclude which patients have PET system saturation based on the sinograms, 3D sinograms created for the first pass bowl reconstruction. If we see any divergent or changes in the sinogram, it might suggest PET system saturation, or at least we conclude that in our EGNM paper. So what effect does the PET system saturation have on the myocardial blood flow estimation? First of all, we observe myocardial PET system saturation in 4 out of 20 patients, or 20% of all the patients when using the full dose protocol, compared to 0% when using the half dose protocol. More of importance here, we can see that the myocardial blood flow obtained using the full dose protocol led to significant increased myocardial blood flow assessments for the full dose scan with saturation when compared to the half dose scan, where we didn't observe any saturation effects. In general, when comparing the Norman plots or test-retest reproducibility as shown here, we were able to observe that there was a bias between the myocardial blood flows obtained using the full dose and half dose protocol. Once removing the data sets with PET system saturation as shown here, we were able to conclude that the bias was reduced by 0.03 milliliters per gram per minute, thus suggesting that the PET system saturation has tremendous impact on the blood flow assessment as a whole. But using a half-dose protocol might introduce a slightly reduced myocardial blood flow assessment. However, in the same paper, we were able to conclude that by using a simple correction factor, as shown here, we are able to preserve the myocardial blood flow assessment using a half-dose protocol as compared to the full-dose protocol. So, in summary, in the 6 papers I presented, there's a common denominator that mainly relates to that motion affects the quantitative accuracy in basically all myocardial PET studies. We can correct for this motion using either data-driven or manual assessments, and by doing so, we can improve the retest reproducibility significantly. We have also shown that injection to scan delay has tremendous impact on the quantitative accuracy when speaking of sodium fluoride PET imaging. And finally, we have shown that using full dose protocols that are currently recommended by the guidelines, we are at risk of having PET system saturation and thereby affecting the myocardial blood flow. By that, I would say thank you for your attention, and I would like to thank everybody here at Cedar who have helped to create this the studies, and also our collaborators that have had significant impact on the sodium fluoride studies.
Okay, excellent presentation. There are several questions from the chat room, so why don't I start with a couple of them. So this is a question related to the blood pool correction. Were you able to plot an increase in the SUV of the plaque with time, as well as reduction in the blood pool signal?
The SUV in the plaque was consistent over a 3-hour over the 1 hour and 3 hour evening protocol. So the uptake in the lesion is consistent. However, the activity in the background dropped significantly, as we were able to show in the paper. Yeah. Okay. Okay. And now for the motion correction, the 3MC work, was the motion correction driven by PET data? The motion compensation was driven by PET data. Yeah. And did you have any issues with background signal from the sodium fluoride? In this context, no. No. Because the motion compensation that we're currently employing focus on the coronary arteries alone, where we use segmentations of coronary CTA scans. Okay. So we're not that driven by the background activity at all. Okay. And how did you deal with the aspect of not having enough statistic for the sodium fluoride dual gated imaging? And how did you reduce the low count bias? So first, how we dealt with the noise. noise per se was not a big issue because we, again, we focus on just the coronary arteries. So once we have data inside the coronary artery and we are within roughly 1 centimeter from the identified lesions, we have quite stable image registrations. So in that sense, noise wasn't that much of an issue. And I can't remember what was the second question.
Oh, yeah, it's okay. Because we have.
Hello, are you?
Hello.
Are you there? Are you
I believe Doctor. Oh, there he is.
I can't hear you anymore.
Yeah, it cut out.
Okay,
Okay, so,
Wunan, it's almost 12 o'clock. Can we close this?
Yeah, so I was going to say, since this is already 12 o'clock,
okay.
Yeah, we're going to end the session so that everyone for their other obligations.
Yeah, I think we should just finish. Yeah.
Yes, great. Well, thank you everyone for attending. I just want to remind you all to take a brief survey. The link is up. And then also stay tuned for next Wednesday session that is going to be on transthyretin amyloidosis from 11 a.m. to 12 p.m. Thank you, everyone.
Thank you very much for organizing this. It was great. Thank you.
Yeah, thank you for the organizer and all the speakers and participants. Yeah, bye-bye.
Bye-bye.
Thank you, Dr. Fayad.
Thank you all. Bye.
Bye.
Visa responder a essas perguntas e que então dão